Cold air is thinning. Ancient ice is cracking. And for the first time in thousands of years, something buried under the Arctic is beginning to move. What happens when creatures that have been frozen longer than human history begin to thaw? What survives after 10,000 years of silence? And what if some of these organisms were never meant to return to a warming world? These questions are no longer the language of fiction. They come from a real scientific concern emerging across multiple research stations in Alaska, Siberia, and the Canadian Arctic. Over the past few years, satellites have recorded sharp increases in melt zones, exposing layers of permafrost that last saw sunlight during the Ice Age. With every meter of ice that collapses, new organic shapes appear. Some are harmless remains, but others still hold trapped cellular life waiting for the temperature to rise. The Arctic is not just melting at record speed. It is revealing a biological archive sealed by nature's most perfect freezer. Microbiologists now compare this region to a time capsule filled with creatures from extinct ecosystems, preserved in a state that modern life has never interacted with. Some of these organisms come from climates very different from the world we know today. If they awaken, what forms will they take? These early observations raise an uncomfortable possibility. When the boundaries between past and present dissolve, the biological rules we rely on may shift. Could something entirely unexpected emerge from this thaw? And if it does, how prepared are we to understand it? Everything that follows will explore how these frozen organisms were preserved, what modern science has found so far, and why their reappearance is reshaping the way researchers think about evolution, extinction, and planetary change. Beneath the melting Arctic lies a sequence of discoveries that scientists are only beginning to piece together. What emerges from this landscape is not a single mystery, but a chain of interconnected clues stretching from microscopic organisms to the remains of colossal Ice Age animals. Each layer tells a different story, and together they hint at a much larger pattern forming beneath the surface. In the next stages of this investigation, the focus will move from the emotional shock of awakening life to the physical processes that allow such preservation to happen. There are cold-driven mechanisms that act almost like natural laboratories, keeping cells intact for millennia and storing genetic information far beyond the lifespan of any species alive today. Understanding these mechanisms is the first step toward making sense of what is now being released. Current research teams are also uncovering fresh evidence that challenges long-held assumptions about extinction and survival. Some organisms appear structurally unchanged despite thousands of years locked in ice. Others show adaptations that do not match any living relatives we know of. What kind of evolutionary history produces this kind of resilience? As scientists examine these findings, a series of unresolved questions grows larger. How many of these ancient organisms can revive? What conditions do they require to become active? And what does their return tell us about the biological past hidden under the Arctic? Each discovery leads deeper into the mechanisms, the evidence, and the limits of current scientific models. The picture becomes clearer only as the complexities accumulate, inviting a more disciplined look into what the Arctic's thaw is truly revealing. The Arctic's ability to preserve life begins with something surprisingly familiar. Imagine placing a piece of fruit inside a home freezer. The cold slows every chemical reaction inside it, halting decay until the moment it thaws. Permafrost works on the same principle, but on a scale far beyond anything found in a kitchen, holding biological material stable not for weeks but for tens of thousands of years. The difference is intensity and time. Temperatures can remain below freezing for entire geological epochs, creating a natural deep freeze capable of stopping almost all biological activity. In scientific terms, this happens because extremely low temperatures limit metabolic processes to near zero. Enzymes responsible for cellular breakdown cannot function, so proteins, DNA strands, and even entire microorganisms remain locked in a suspended state. Researchers describe this as cryopreservation, a process in which life is not dead but simply paused until conditions allow movement again. The permafrost acts like a stable chemical vault, keeping cells isolated from oxygen, heat, and microbial competitors that would normally destroy them. One of the most intriguing mechanisms is vitrification, a phenomenon where water inside cells becomes glass-like rather than crystalline. Crystals tear through membranes, but vitrified water preserves structures without causing damage. Some microscopic organisms, especially ancient nematodes and unicellular species, appear to have mastered this survival strategy long before humans even existed. Their bodies shift into a dehydrated state, allowing them to withstand cold that would destroy complex life forms instantly. When warmth returns, metabolic activity slowly resumes, raising the question of what other species may have evolved similar protective strategies. These processes also interact with geological rhythms. Layers of permafrost form and deepen during long glacial periods, embedding organisms in a vertical timeline. Each layer corresponds to a different climate, atmosphere, and ecosystem. When the Arctic warms, these layers melt from top to bottom, releasing organisms in reverse chronological order, from relatively recent species to those that lived alongside mammoths. What does this upside-down timeline imply for modern ecosystems? Another mechanism at play involves ancient soil chemistry. Low oxygen levels reduce the presence of free radicals, molecules that typically damage DNA. Without oxygen and without heat, genetic structures remain unusually stable. Some ice cores show DNA strands so intact they resemble samples only decades old. This suggests that life from climates very different from ours may be returning with minimal structural decay. These mechanisms create both scientific opportunity and uncertainty. If the permafrost functioned as a perfectly controlled environment for millennia, then what emerges from it may be better preserved and potentially more active than expected. It invites debate. How many forms of life can survive such extreme dormancy, and what does their survival say about the boundaries of biology? Recent excavations and melt zones across the high Arctic have produced a stream of observations that deepen the mystery. What once seemed like isolated findings now form a pattern woven through biology, geology, and climate science. These observations are not speculative. They come from field reports, ice core analyses, and documented laboratory revivals that researchers have repeatedly verified. Each point adds weight to the idea that the Arctic is releasing life far older and more resilient than expected. One of the clearest observations comes from the revival of ancient microorganisms. In Siberia, researchers successfully reactivated microbes estimated to be roughly 10,000 years old, allowing them to metabolize again under controlled conditions. This revival is not theoretical. It has been reproduced in different laboratories, demonstrating that some species can endure complete metabolic shutdown for periods longer than recorded human civilization. The fact that these organisms restarted biological functions so easily challenges our assumptions about the limits of cellular survival. A second line of evidence comes from the discovery of cold-preserved multicellular organisms. Recently, nematodes extracted from permafrost layers more than 30,000 years deep showed signs of life when slowly warmed. Although these organisms do not reawaken in the dramatic sense popular media often imagines, their ability to resume basic activity after millennia of silence suggests that biological stasis can operate at scales previously considered impossible. This observation forces a reevaluation of how life persists in extreme environments. A third observation emerges from the condition of larger ice age remains. Mammoths recovered from melt zones often retain soft tissues, detectable proteins, and in some cases intact hair follicles. While these animals cannot revive, their preservation reveals how effectively the Arctic's cold controlled decomposition. Some tissues appear so structurally intact that they have become valuable for studying ancient pathogens that lived alongside these animals, organisms that modern ecosystems have never encountered. This raises questions about the biological history locked inside such remains. A fourth observation involves the chemical stability of ancient DNA. Genetic samples extracted from deep permafrost show remarkably low fragmentation, with some strands almost comparable to modern samples in length and clarity. Such preservation is rare in most environments, where heat, oxygen, and microbial activity rapidly degrade DNA into unusable fragments. This level of preservation allows scientists to reconstruct species that never left fossils in traditional archaeological sites, revealing lineages previously unknown. A fifth observation comes from satellite and climatological monitoring. Data from recent years show a measurable shift in the physical structure of permafrost layers. As upper regions thaw, meltwater penetrates deeper, destabilizing layers that have remained untouched since the last glacial period. Thermokarst formations, sudden ground collapses caused by melting ice, have become more common, exposing new biological material at a pace faster than researchers can analyze. This acceleration creates both opportunity and scientific pressure, as each collapse may reveal organisms older than the last. Taken together, these observations suggest a landscape undergoing rapid transformation. Some findings provide clarity, while others raise new uncertainties about what the Arctic's deep freeze has truly preserved. The evidence is accumulating faster than explanations can keep up, pointing toward a biological archive that is still far from fully understood. One interpretation of the Arctic thaw suggests a straightforward story. Organisms froze, remained inert, and are now returning as passive remnants of the past. But science rarely settles on a single clean explanation, especially when the evidence comes from environments as complex as permafrost. Several alternative models compete to explain what is truly happening beneath the ice, each offering a different picture of how ancient life persisted and what its revival actually means. A widely discussed alternative is the contamination model. According to this view, some samples recovered from deep layers may not be genuinely ancient. Modern microbes sometimes infiltrate permafrost through microscopic cracks or meltwater channels, colonizing spaces that appear ancient but are chemically accessible. This model works well for organisms that show metabolic activity too quickly or display genetic signatures identical to modern strains. However, it struggles when revival occurs in strictly sterilized conditions using samples extracted from sealed cores with no clear infiltration path. Another explanation centers around cryotolerance rather than deep-time survival. In this model, the organisms found in thawed layers may not have lived continuously for tens of thousands of years. Instead, they could have survived by entering cycles of near-death dormancy during brief warm periods before being refrozen again. This mechanism is plausible for microbes adapted to fluctuating polar climates, but it cannot account for multicellular organisms preserved in layers far older than the cycles themselves, where geological evidence shows uninterrupted cold. A third hypothesis emphasizes chemical preservation rather than biological resilience. Here, the idea is that some revived organisms may not have persisted as whole, intact life forms. Instead, permafrost may have preserved fragments of cells, proteins, and genetic material that later reassembled or interacted with modern microorganisms in laboratory settings. While theoretically interesting, this model faces difficulty explaining nematodes and other multicellular species that resume activity without requiring reconstruction. A more conservative model proposes that some revived life may represent rare outliers rather than widespread ancient survival. In this view, a handful of species evolved unique mechanisms that allowed them to withstand extreme cold, while countless others simply perished. This hypothesis fits the biological record, which often favors resilient specialists during environmental stress. Still, it does not fully explain why preserved samples across distant Arctic regions show similar patterns of cellular stability. A final alternative attributes many observations to ecological ghost signals. This refers to measurable biological traces, DNA, proteins, lipids, that survive long after organisms themselves have died. Researchers may interpret some signals as evidence of revival, when in fact they reflect chemical persistence rather than biological activity. This model is valuable for interpreting DNA stability, but it cannot replace direct observations of organisms that clearly resume metabolic functions. Among these explanations, the simplest and most consistent with current evidence is the deep freeze preservation model, supported by stable temperatures, minimal oxygen, and cryoprotective cellular structures. This model does not claim that all thawed organisms survive, nor that every revival reflects ancient biology. It merely aligns with the physical and biochemical conditions documented in Arctic cores, offering the cleanest fit to the currently available data. Despite its strengths, even this model leaves room for debate. Each alternative illuminates blind spots in our understanding, reminding us that ancient life emerging from ice is not a single story, but a set of overlapping possibilities still being tested. Large sections of the Arctic remain unknown not because the evidence is missing, but because the conditions under which ancient life survives are more complicated than any single model predicts. One of the biggest gaps lies in understanding the true limits of biological dormancy. Scientists can document revived organisms and analyze their cellular structures, but they still cannot explain with certainty how long life can remain inactive without losing viability. Laboratory experiments simulate cold, dehydration, and chemical stability, yet these simulations cover years or decades, not tens of thousands of years. The scale of natural preservation far exceeds the timescales we can experimentally reproduce. Another uncertainty comes from the internal chemistry of ancient cells. We can extract DNA, measure protein integrity, and observe metabolic signatures, but we cannot fully determine what subtle changes accumulated during millennia of stasis. Some revived organisms behave normally, while others show unusual metabolic patterns that do not match modern relatives. Are these differences signs of ancient adaptation, or simply the result of cellular damage too small to detect? The line between genuine evolutionary traits and long-term chemical distortion is still unclear. Geological questions complicate the picture even further. Permafrost layers are often described as stable, yet subtle movements caused by freeze, thaw cycles, shifting sediments, or meltwater infiltration can disturb their structure. This means that some samples labeled ancient may have been displaced from their original positions. Without complete certainty about a layer's stability, estimating the true age of embedded organisms becomes a challenge. Even high-resolution dating methods carry margins of error that widen as samples approach the limits of known geological timelines. There is also a gap in knowledge regarding the ecological consequences of revived organisms. Most laboratory studies take place under controlled conditions, where revived life is isolated and monitored. But real environments contain competing species, fluctuating temperatures, and chemical pressures that no laboratory can perfectly recreate. A revived organism that behaves harmlessly in a laboratory may act differently when returning to a dynamic ecosystem. Even basic questions remain open. How quickly could such organisms spread? What conditions would limit them? And how would they interact with existing species? Another major unknown concerns genetic stability. Ancient DNA appears well preserved in many cases, but preservation does not guarantee completeness. We often recover fragments that represent only a portion of an organism's genome. This means our reconstructions may be accurate only to a point, leaving entire biological functions unobserved. Some revived organisms could rely on genetic mechanisms we do not fully understand, especially if they evolved during climatic periods unfamiliar to modern ecosystems. A final area of uncertainty lies in the speed and scale of Arctic thaw. Climate projections vary widely depending on regional patterns, atmospheric models, and feedback loops involving methane release. If thawing accelerates faster than predicted, thousands of biological samples could surface before researchers have the capacity to study them. The sheer volume of emerging material might outpace scientific analysis, leaving large portions of it undocumented or poorly understood. These unknowns reflect the complexity of studying life that bridges past and present. Even with careful research, the full picture remains incomplete, shaped by gaps in our tools, our timescales, and the environments we can replicate. Understanding what the Arctic holds requires not only data but patience, because the most important details often lie just beyond the reach of current knowledge. When ancient ice finally releases the life it has held for ages, the central questions return with greater clarity. Can organisms truly survive tens of thousands of years in complete dormancy? What kinds of species are emerging from the thaw, and what do their reawakened forms reveal about biological endurance? And most importantly, do these discoveries represent a threat, an opportunity, or simply a mirror reflecting the planet's deeper evolutionary history? The evidence gathered from multiple research stations suggests that survival on these timescales is possible, though not universal. Microorganisms show the highest success rates because their metabolisms can slip into states so slow that biochemical decay becomes negligible. Some Arctic species employ natural cryoprotectants that prevent ice crystals from forming inside their cells, while others dehydrate themselves into a glass-like state in which internal structures remain intact. These mechanisms allow them to resume activity once temperatures rise, offering a clear explanation for documented revivals in the lab. For multicellular organisms, the story is more selective but no less remarkable. Nematodes recovered from deep permafrost have demonstrated the ability to rehydrate, repair minor molecular damage, and begin simple movement again. Their survival likely depends on extremely low metabolic requirements and specialized proteins that stabilize tissues during prolonged freezing. While larger animals cannot revive, their soft tissues preserve clues about the pathogens, parasites, and microbial communities that once inhabited Ice Age ecosystems. These remnants help researchers reconstruct ancient biological networks and understand how environments shifted during glacial cycles. Concerns about potential danger often arise when unfamiliar organisms resurface. So far, revived species are slow-growing, sensitive to oxygen, and require precise conditions to remain active. Their abilities do not resemble fast-spreading modern pathogens. Yet, the presence of ancient viruses preserved in permafrost adds a layer of caution. Several samples have shown that frozen viruses can infect host cells under laboratory conditions, though none have demonstrated viability outside controlled environments. This suggests the risk exists but is limited by environmental constraints and the delicate nature of these ancient forms. Ultimately, what emerges from the Arctic reveals a complex but grounded reality. Some organisms can endure extraordinary spans of time. Their return does not signal an immediate biological threat, but instead expands our understanding of how adaptable life can be when placed under extreme conditions. The thaw exposes systems that were once sealed, offering insights into evolution, climate history, and the boundaries of biological survival. These answers show that the Arctic is not unleashing monsters from the past, but rather releasing data, biological, chemical, and environmental, that helps refine our understanding of the planet's long memory. The melting Arctic is more than a scientific event. It is a reminder that time is layered, that the past is never as distant as it feels, and that the planet preserves its own history even when we forget it. The organisms emerging from the ice are not messages of fear or hope but fragments of an ancient world returning to the surface. They challenge the belief that life operates on predictable scales and remind us that biology has always been more flexible than our definitions allow. These discoveries carry a broader meaning. They show that the planet stores information in forms we rarely consider, and that climate shifts can reveal knowledge long hidden beneath the surface. Every revived organism, every well-preserved strand of DNA, becomes a lens through which we understand not only the past but our own place within it. The thaw forces us to reconsider what it means to coexist with a world that is constantly changing yet constantly returning pieces of what it once was. There is a quiet lesson in all this. Humanity tends to view history as something written exclusively in books, fossils, and artifacts. But the Arctic teaches a gentler truth, that history can also be living, dormant, and waiting. Its return is not a threat but an invitation to observe carefully, question deeply, and engage with the planet as a dynamic record rather than a static backdrop. In the end, the significance of these discoveries lies in how they reshape our understanding of resilience. Life endured ice ages, extinctions, and planetary transformations long before humans appeared. To witness its reemergence is to be reminded that the world is far older, stranger, and more interconnected than our daily concerns suggest. Understanding this complexity allows us to view the Arctic not as a distant wilderness but as a library of deep time slowly opening its doors. Thank you for listening to this exploration. If this kind of scientific storytelling resonates with you, consider subscribing to the channel to follow future investigations and discoveries.